Hello, my name is Andrew Roulard, and this is the fourth part of the lecture on resources for bioinformatics and machine learning. The next resource is called the Human Endogenous Complexome. This is a high-content proteomics dataset consisting of 3,000 immunoprecipitations followed by mass spectrometry. You can access this data through this website shown at the right, which is called the Nuclear Receptor Signaling Atlas. And so what this dataset is telling us is each of these immunoprecipitations is isolating a particular protein, and along with it, any proteins that are in a complex with that particular protein. So each immunoprecipitation provides a picture of the proteins that are interacting either directly or indirectly in one or multiple complexes with this one protein that we were trying to isolate. So this is a useful data set for finding functional protein-protein interactions. This next slide is showing how this data can be organized. This format is known as a set library. Each row is reporting a set of genes. The first entry in the row is indicating a particular immunoprecipitation experiment and also identifying the particular protein that was isolated for that immunoprecipitation. And then the rest of the row is listing out all of the other proteins that were recovered in this complex that was pulled down in this particular experiment. So on this next slide, we see when we organize this data into a matrix format, we can see how proteins are associating with each other in these various complexes. The next resource is called the Online Mendelian Inheritance in Man, or OMIM for short. You can access this resource by going to this webpage shown at the right. OMIM is a database of human phenotypes, mostly diseases, that have a known genetic basis, and it's summarizing what's known about the phenotype or the disease and the genes that are involved. The simplest abstraction of the data would just be a set of genotype-phenotype associations, which can be downloaded to understand genes that play a role in these various phenotypes or diseases. This is another one of these formats that's kind of like a set library, where in one column we have listed diseases or phenotypes, and then in the next column we have a set of all of the genes that are known to have mutations associated with these diseases. So again, we can reorganize this information into a matrix format and plot it as a heat map and get this broad overview of how gene mutations are related to a large number of human diseases. The next resource is called Proteomics Database. This is a website that's hosting a dataset that profiled protein expression in a large number of cell lines and tissues. You can access this data through the website shown on the right. And this is one of several proteomics resources. A similar dataset is called the Human Proteome Map, which also is a project that measured protein expression in a number of cell lines and tissues. So this is very similar to gene expression data that we discussed previously, except now instead of quantifying expression of genes, the dataset is quantifying expression of proteins. This next slide is showing what this dataset looks like. Again, we have the data organized into a matrix format. Each column corresponds to a different cell line or tissue, and the rows correspond to particular proteins, which in this case their proteins are mapped to gene symbols. And then the matrix values are reporting the abundance or expression of the proteins in these different conditions. So this data set can be converted to differential expression scores and then thresholded to extract the significant protein cell line or protein tissue associations. This next slide is showing the heat map that results when you do this analysis to compute the proteins that are differentially expressed in these cell lines or tissues. And you get again a broad overview of how proteins are related to each other based on their expression in a large number of cell lines or tissues. The next resource is called the Allen Brain Atlas. You can access this resource through the website shown on the right. The Allen Brain Atlas hosts a number of brain-related data sets, including detailed anatomical maps of the mouse brain and connectivity maps where projections of neurons are mapped throughout the brain. And what I'm going to talk about is whole genome gene expression profiling, which is measured in very, very high resolution throughout the mouse brain. So this resource can be useful for mining for genes that are differentially expressed in a region of the brain or at a developmental time point for the time series data. This next slide is showing what the data looks like. Again, we have essentially just a gene expression matrix like we've seen before. Columns correspond in this case to particular brain regions, and the rows again are genes, and the values of the matrix are the expression level of the gene in these different regions of the brain. So we can convert these absolute values of gene expression to relative values and get these differential expression scores, and then threshold our matrix to extract just the significant gene brain region associations. The heat map on this next slide is showing you what this matrix looks like. This is connecting genes, which are labeling the rows, to brain regions, which are labeling the columns. The red regions on this heat map are indicating brain regions that show significant upregulation of genes, and the blue regions are showing brain regions that show significant downregulation of a group of genes. So again, this data set just provides another perspective on how genes are related to each other, in this case, based on how their expression varies over a number of brain regions. This next resource is called the Genotype Tissue Expression Project, or GTEX for short. This is a genome mapping project, and the goal of the project is to discover expression quantitative trait loci in the human genome. So these expression quantitative trait loci, or EQTL, are regions of the genome where variation in the DNA sequence is correlated with variation in the expression of some other gene or a set of genes. So this is a useful data set for trying to understand how variations in individual genomes impact the expression program of cells in a particular person. In the next slide, we see what the data looks like. Here we have just a simple list of associations. One column is reporting genes, and another column is reporting genomic locations. In this case, those are encoded in what's called a SNP ID, but these SNP IDs are associated with a particular region on a particular chromosome in the human genome. So we can parse this data set and reorganize all these gene SNP pairs into a matrix, as shown on this next slide, where we have, again, genes labeling the rows, and then these SNPs, or these genomic locations, labeling the columns. And again, this is just another piece of information telling us how genes are related to each other, in this case based on their connections to these SNPs that could be regulating their expression.